Hi everyone. Before we start this week's webinar, I have to make an announcement. December 1 is a public holiday in the UAE, so we will not have a webinar that week. We'll meet next week, which will be on December 8th. Today, we have reached an important milestone. This is webinar number 20th of our series. And so we have a special guest, Axel Brandenburg from Nordita. Nordic Institute of Theoretical Physics from Stockholm. He works in the field of astrophysical fluid dynamics. His main interests are concerned with magnetic field generation from turbulent motions with applications to the sun and stars, accretion disks, galaxies, and the early universe. He has made numerous contributions to the field. He's also a distinguished fellow of our center. One of his interests is also astrobiology, and he will talk about a very interesting paper he has written recently. So, Axel, welcome to the center in this virtual mode, and let me hand it over to you now.
Thanks, Dimitra, for your very kind introduction, and thanks to both you and Srini for inviting me. It is really a pleasure to be associated with the center, and I enjoyed the visit of January earlier this year very much. Here you see the cup with the symbol of the center, of course. So this was such a wonderful time. And it's such a shame that I cannot be with everybody at the center at the moment, but I really would like to come again and stay there for longer. Today I will be talking about a subject. It's a little bit of a hobby of mine, homochirality. There is some connection with my with my other work on astrophysical fluid dynamics, but it's actually rather remote. And I really came into this just because I was interested in astrobiology. So therefore, I will also begin this talk by telling you a little bit about the history in our search for life. Next, I will come to the question why chirality is actually important for this question. How do we can envisage amplification mechanisms of chirality, the single-handedness of all our biomolecules? You see here an example of two different hands, one left and right hand, and only one of the two amino acids is actually realized in actual life. The question is, how is it possible? Is this because of certain external factors, like, for example, the weak force in theoretical physics? Or is it because it is actually an amplification mechanism that arises as a consequence of life? And then finally, I will end by talking about possibilities of extinct and extant life on Mars, and perhaps the possibility of detecting chirality. As I said earlier, this is a little bit of a hobby. I had early works in the 2000s on that, but most recent work was of 2019. Astrobiology really is something of the last 20 years, and it rests on four major cornerstones that really made it a big science on its own, even though it doesn't actually have real data for life on other worlds other than the Earth. One of the cornerstones is the realization that back in time, we have evidence from the deficiency of carbon-13, which is a marker of the possibility of life relative to marine carbonates, that this difference has extended into the past by almost 4 billion years. And that is so early on that one really must think that life may have been already possible immediately once the Earth became cold enough and we had stable continents. Another important realization is from the hydrothermal vents and the realization that very deep down in the ocean, life can thrive, and it thrives by means of chemical energy and not photosynthesis at all. And so it's a very different type of life, and that also led to the idea and the discovery of different life forms, like, for example, the archaea, that have not been known in the time when Martian exploration began. Concerning Mars, is the possibility of water on Mars, even at mid-latitudes and certainly at high latitudes, has been realized over the last 20 years almost. And also the idea that planets other than our own Earth exist in numerous amounts. In particular, an exciting example is Proxima Centauri, the nearest world of an M dwarf nearby the Earth. So astrobiology has really emerged in the last 20 years as an important science. And one of the important realizations is that the possibility of microbial life may be extremely universal and could perhaps exist everywhere. We just have not found any examples so far. But it is really amazing that we do know of life forms nowadays that can exist under unimaginable conditions, even perhaps in the atmosphere of Venus is one of the latest recurrent ideas. There's also the possibility of having an independent genesis of life, so life which is then really originates completely independent of our Earth life, and Mars may be one such example. This one of the important differences for astrobiology relative to the previous science of exobiology is really that astrobiology does include the study of life on Earth as well. And this is what has made astrobiology such an important science these days. Just to put the scene here between what we nowadays think that of the rare Earth hypothesis, which means microbes are really a universal possibility everywhere, relative to what people thought in the past, that we may even have intelligent life on Mars. This is an idea, of course, here, the interesting maps. I mean, these are actually highly accurate maps of Schiaparelli with very accurate longitude determinations that are still accurate to within a few degrees. But the real killer to the idea of intelligent life really started with Mariner 4 back in '64, when it was realized that the first pictures from Mars really looked like almost the moon, and there was no possibility of, there was very, very thin atmosphere only, and certainly no intelligent life. However, it is really not so far-fetched, and one has to understand why people have been thinking about this. I mean, even just look at the pictures from this paper by Herschel, where he realized that a very thin but moderate atmosphere does exist, so that its inhabitants, he wrote in his paper of 1783, probably enjoy a situation in many respects similar to ours. So it really makes us aware that the idea of intelligent life was just so uniformly accepted. So we must be somewhat forgiving to the people who make jokes about this work of Schiaparelli, for example, and not really Schiaparelli so much as others since then. So Mars really was an environment which was most likely warm enough to have liquid water at its surface for at least 1 billion years just after its creation. And we see here many evidence of flowing water in the past. So it's most likely an environment where extinct and perhaps even extant life may exist. One of the realizations from the Earth, again, is that life can not only thrive, but it may have actually started in the proximity of hydrothermal vents. These are systems which are very rich in terms of chemical energy. They have low pH values, high pH values, as opposed to the so-called black smokers, which are acidic. So alkaline vents are most likely the sites where we have hydrogen and methane, both very important energy sources for early life. And there are ideas even of having primitive cellular cells, actually, that consist of the precipitates of iron sulfide, pyrite, in the proximity of these vents. So you may have, therefore, from these precipitates, new little compartments that could be in the early sites of life without even having any cells. So these compartments, these very small compartments, might be actually the sites where life could begin and also develop through Darwinian evolution. So let me now come to the question of handedness. I will say in a moment why it's important, but let's first understand what it actually is. It is a characteristic that is very closely connected with the fact that carbon has a valency of 4. So it's 4 legs, therefore, may either connect with the same or with different other compounds. Here in this case, we see the example of an amino acid, which consists of a COH group and an NH2 group. And here in this particular case, we have 2 of the other compounds being the same. However, it is possible that they are not the same. So that's what I meant. These are here the same. And therefore, if you look at this in a mirror, it looks the same. Unlike this molecule here, where all 4 are different. All the 4 residuals are different. And therefore, it looks different if you look at it in a mirror. And this was an important realization that goes back to Louis Pasteur back in the 17th century. It also, and that was also an important realization by Pasteur, it also is able to make itself evident through the fact that it can have an effect on polarized light. In fact, we do have 2 different forms. They are called D forms for usually dextrorotatory, and the L form for levorotatory. So they rotate polarized light, which you produce by having a polarizer on unpolarized light. This makes the light polarized. Then you put your substrate in between, and it rotates the plane of polarization in a certain sense, either right-handed, in which case you have the D form, or left-handed, in which case you have the L form. For chemists, of course, they may, anyway, that's so important. So this is an important distinction between the 2 different types of biomolecules. The realization is really somewhat in more modern times. For example, one of the famous examples where it was not realized, where its importance was not realized in the production, in the synthetic production of medication. Here, for example, the medication thalidomide, which was used in order to cause, in order to cure to cure morning sickness during pregnancy. But it also, when you have it in the wrong form, both of them have in principle similar functions, but the one of the wrong form led to misformations of people that were born around that time. So people around the time when I was born, many of those were actually born with misformations because this was thought to be a safe medicine at the time. Nowadays, it's realized that it was not safe. Any non-biogenic formation, production of biomolecules, and here, for example, is this famous Miller-Urey experiment, where the first, where one was able to produce in the presence of lightning and electric discharges and heat, one was able to produce something like 15 different organic compounds, including amino acids, 11 different ones. But all of those come in either of the two, in both of these two handednesses. It's called a racemic mixture of molecules. So we do have molecules that have a handedness, but you have equally many of the right and the left-handed ones. So now comes an important experiment. This was done by Joyce and including Ogle in the year 1984, where they studied what is known as template-directed polymerization or oligomerization. These are short molecules, and their lengths can be measured by chromatography. Chromatography is something where the lighter ones can rise on a strip of paper more rapidly than the heavier ones. And so you automatically get a histogram of the lengths of different polymers or oligomers in your substrate. And so if you have a so-called homochiral environment, every single monomer has the same handedness, then you are able to produce on a template, which is one of an RNA molecule where the other side has been, or a DNA molecule where one side has been stripped, it can act as a template and can then attach monomers to it, as very much as it is now used nowadays in all the polymerase chain reactions, PCR reactions that are being used to amplify DNA. So we can grow these polymers in the absence, however, in this case of any biological catalyst. However, if you attach monomers of only the opposite handedness, polymerization cannot grow. So the polymers remain extremely short. This is because of something like a poisonous effect of a monomer of the wrong handedness on an existing isotactic polymer, a polymer that has the same handedness throughout. If you then begin to attach a monomer of the opposite handedness, polymerization comes to a halt. And this was realized to be a major problem to the idea of selecting only one of the two handednesses. If you have a mixture of not just the right ones, but also the wrong ones, polymerization stops. That's the difference between these two experiments. Here was just monomers of the wrong handedness. And in this experiment, you have a mixture of the right and left-handed ones. Polymerization extends up to maybe 6 monomers, but then it really comes to a stop, unlike the case of an homochiral environment. How is it possible? Let me contrast this with a very simple chemical reaction for life. And of course, this is just sugar here, but basically all photosynthetic life is of something like that form. So life can be really thought of something like some types of sugar molecules that are synthesized from, and they can be chiral, they can have a handedness, and yet they are synthesized from compounds that are not only completely achiral, they are even completely non-biological. So here we have just water and we have CO2. And this is what leads to the formation of plant life in the presence of photosynthesis. In that case, it is actually the chlorophyll that acts as a catalyst which introduces and allows only for one of the chiralities. So this is how life, contemporary life, can do it and produce, therefore, sugars that have one particular handedness. But this is not possible in prebiotic environments because we simply did not have any catalysts like, for example, chlorophyll, which already had imprinted its single chirality at the time. So therefore, we have to think about completely different mechanisms at the moment of the origin of life. So life would not have been possible to even really get started or to develop if there was no such possibility of having a selection of one of the two chiralities. So this leads us to the question whether the life could have possibly started at all in an achiral environment. In fact, there are some biologically significant molecules that can be completely achiral. So DNA is our current information carrier, but there were other molecules like peptide nucleic acids, PNA, that can, in certain circumstances, be made completely achiral. So this is indeed one possibility. However, there's also the possibility that certain physical factors, we must discuss this possibility, physical factors like, for example, the possibility of polarized photons, polarized light, could lead to the presence of only one of the two chiralities. I should warn you already now that any of these mechanisms is likely to be a very weak one. It would not lead to a full 100% chirality. And it's therefore important to think of the possibility of at least some amplifying mechanism. And this is indeed a possibility that in the presence of two important mechanisms, one is autocatalysis, that is making more of itself, can be an important ingredient during the polymerization. But we need another one, and that one is called, I come to that in a moment. Before coming to that, let me explain briefly how it is indeed at all conceivable that an external influence can produce handedness. And this is related really to a recent to work of the 50s, when it was realized that one of the fundamental forces of nature, the weak force, actually itself already exhibits one particular handedness. It's therefore a non-mirror symmetric force. An important experiment would be if you take a neutron and let it decay. A free neutron decays in 10 minutes. And it would decay into a proton and an electron and an anti-electron neutrino. And these electrons all have a particular handedness in the sense that it has a spin which is either aligned with its momentum or anti-aligned with its momentum. And now we can ask the question, which of the two possibilities do we actually have in nature? And it turns out that this decay of a neutron, which is called a beta decay, it only produces electrons of one particular handedness. So this was realized back in 1956, and it may have also some bearing on the production, or rather the destruction, of one of two possible polymers of life. Nowadays, most of our life consists of a spiral, which is a right-handed DNA spiral, and it has its origin, of course, in the fact that also the sugar molecules, which are part of the backbone of the DNA molecule, is right-handed. However, there could have been a selection effect. In an early prebiotic world, you might have molecules of either the form that we do have in contemporary life, or the opposite. And this is the word life being spelled backward. This is what we don't see. And polarized light or polarized photon, highly energetic photons that could emerge from the decay of cosmic radiation would have a preferential destruction effect on the polymers and monomers of the wrong-handedness. This is one possibility. However, we do need an amplification mechanism, and I want to briefly explain how this is at all conceivable. And it was an important realization that goes back to the work of Charles Frank of 1953, who produced a very simple idea. And this idea has since then been tried to verify in various in various settings that may be to various extent more or less astrobiologically relevant. Many of them are also not directly relevant, but the idea is certainly very interesting. And the idea is that you do need two mechanisms. I mentioned already autocatalysis, but another important mechanism is what is known as mutual antagonism. And what it was at the time was not clear. However, the mechanism that we have seen a moment ago in the paper by Joyce et al., where only one of the two polymers can grow, namely the ones which produce polymers of the same handedness, this is one such mechanism, which would be a mutual antagonistic mechanism, which would have a preference of giving preference to those that are already in the majority. If you consider two molecules with a slight difference, and this slight difference could really just come about because of statistical fluctuations, either locally on the Earth or locally, and certainly you would have always a location where one of the two handednesses would be more preferred, would be slightly more than the other one. But which of the two is at this point not necessarily very clear, unless, or not very important either, unless, of course, you are interested in the idea of explaining why exactly our life was left-handed. It had left-handed amino acids and right-handed sugars. In principle, it could have been the other way around. And this is one of the important questions when we look for extinct or extant life on Mars, which would be an idea for a completely different genesis of life. So if we have a small excess of one of the two other ones, then what mutual antagonism actually really is that it destroys an equal amount, not an amount that depends on the majority, but actually an equal amount. An equal amount of the same. So you have the destruction of one against another. So it's a one-to-one fight. One kills another guy, and it continues until only one of them is left. I have here another example where you see the slight excess, and each time you delete is the same amount until eventually you have 100% excess. At this point, you have it at the expense of the length of the amount of polymers. Only a very small amount of polymers is left here. That is why you also need autocatalysis. But autocatalysis would lead to an amplification of not only this, but both of them. So these two mechanisms are completely agnostic regarding which of the chirality it would amplify. But what it would do is it would amplify one of two possibilities in the end. So here's the example shown where we have not only antagonism alone, but also combined with autocatalysis. That means at each moment we stretch the two columns, both columns, by the same amount. You stretch it here also by the same amount, and even here you stretch it by the same amount. But by this moment, if you do it for the last time, if you have again the destruction of equally many and stretch it again, you have now an equal amount of polymers of the same handedness that were initially only in a slight excess. So this is an amplification mechanism that may be realized in various biotic systems. One of the possibilities in an early RNA world. An early RNA world is an idea that goes back to the 60s when it was hypothesized that life may have started through an information carrying molecule rather than a molecule that was just doing just feeding on itself and replicating itself, but actually a molecule that was also able to contain information and therefore lead to eventually Darwinian evolution. So these were rather simple ideas, and one needs to go further than that. Several important steps could lead to more realism of these very early ideas. One of them is the idea that polymerization should really be carried out, or one should really study this idea of Frank, which was simply just on single molecules. One should really extend this model to polymers. That was not done in his early work. That has been done subsequently. Another important mechanism could be to consider not just these more complicated RNA molecules, but already amino acids, peptides, which are polymers of amino acids. This would lead, this could also lead to a selection of one of the two handednesses and would lead to a prototype of a mechanism that could select one of the two handednesses. And the last extension of the model is to consider more spatially extended molecules, extended worlds, where you could have one handedness being produced in one part of the Earth and the opposite handedness in another part on the early Earth. So this could lead to different chiralities. And then the question is, which of those two would then finally dominate? So let's come to the first step, where we have polymerization. This was a model that was considered in a paper by Sanders of 2003. So all this is rather work of the last 20 years or so, where he considered a simple model where a polymer of the same handedness can grow if it has the same handedness, if an L polymer attaches to an L left-handed monomer. That would produce a longer polymer. So this can lead to chemical reaction equations that could be solved like an ordinary differential equation. And then you have a similar reaction for what is known as enantiomeric cross inhibition, the mechanism that was found in the work by Joyce in his 1984 paper. I showed you the chromatographs from his work. If you have a polymer of one-handedness and you attach to it a monomer of the opposite handedness, here L and R refer to left and right, then you would produce a new polymer, but further polymerization of this would not be possible. And so this can be modeled by having a polymerization coefficient that is then different from the one for isotactic polymerization. This is one idea. Another idea is to work with a peptide world. In that case, instead of working with an RNA world, you would work with peptides. And there is an interesting idea that goes back to the work of Poisson of 2004, which does not involve at all any polymerization. It involves instead activation, polymerization, and then an important mechanism is what is known as epimerization. Epimerization leads to the change, the spontaneous change of one of the two handednesses in a short polymer. And here, in this case, we have a mixture of a D and L, dextro and levo rotatory, that can lead to the change of one of the two. And in particular, it's the one on the right, only the one on the right that can stay, and the one on the left would spontaneously swap its handedness. This is a process that is called epimerization. And again, it's completely symmetric, this model. But solving, and then finally you have the process of depolymerization, again, completely symmetric everything. And you can go in a circle and produce in the end an amplification. And finally, we have the possibility of a spatially extended models. I myself have been working on this already back in 2004, on this and also the polymerization models. And it leads to interesting analogies with the spread of diseases at the time. This was if you go to the important textbooks of what is known as reaction diffusion equations, in a spatially extended system, you have your local ordinary differential equation mechanisms, here a very simple model of this, But then you have a diffusion term, both equally symmetric for the right and left-handed forms. So this goes completely analogous to what was done to model back then the spread of the Black Death. Now we have another disease, of course, and so I'll come to this also in just a moment. It leads to interesting ideas where the spatial propagation of a disease happens with a constant speed. And so that's also an important realization back then. If we have, however, the Black Death and the White Death as the two different life forms, both of them can grow through the combined process of autocatalysis and enantiomer cross inhibition or mutual antagonism. It can spread in both directions in an abiotic environment. However, once two different life forms of opposite handedness come in contact, it leads to a front that can no longer propagate. It stalls. And so in this particular simple case, there would be no possibility of having one of the two handednesses finally dominant. So one must ask the question, how was that even possible on the early Earth? Now, there's a simple answer to that. If you actually solve this model in a more than just one-dimensional model, you see a surprising effect here. You see that we have different black and white diseases, so to speak, or early life forms. And you see also always that the rings of one particular life form that are here in the minority begin to shrink. If I continue this model, you see these different ones begin to shrink. This was a short run here. We see the same phenomenon in a much longer time scale. And you see that eventually only one of the two handednesses will survive. This is and so now you see the last one will shrink, and then you have only one of the two handednesses. Why is that possible? Consider simply so why is it possible that it always shrinks? And the reason for this is that if you count the number of soldiers or molecules that fight against the other ones through mutual antagonism, then if you put them all in a row in a circle like this, and you put soldiers of the opposite handedness or soldiers that again fight against them on the periphery, on the outside, they are always in the majority, but not by much, in fact, only by 3. This has to do with the number of pi. This does not change at all. This is remarkable, even if you take many, many more soldiers. Again, and that's because they are then closer together. So here you have 120 on the inner circle and 122 on the outer circle. The difference is always just 3. But there is a difference, and so this leads to the shrinking of this molecule on a slow time scale. And so we have estimated that even on a prebiotic world, you could have maybe maintained one of the two handednesses for up to 500 million years. That's not long enough, so we should not expect to have a homochiral environment within very early times. Maybe on Mars, there may still be evidence for this, but on the Earth, again, it would be too late, most likely. So now let me come to an update about the coronavirus. In fact, it displayed a remarkably regular behavior where the number of infections was not growing exponential at all, not even in the early times, not even after January the 20th. That was basically all restricted to the city of Wuhan back then. So a simple way of explaining this is by very much the same mechanism, where you have a handedness, where you have the disease, and it can only spread through the interaction on the periphery of an already infected population. And this is what can, by very simple arguments, explain a simple quadratic growth, simply because the growth is proportional to the length of the periphery and not proportional to the total number of infected. So this is a bit different from the standard model, but it can in fact, and this is a recent paper here of mine in the infectious disease modeling journal, it can indeed lead to explain the quadratic growth. And the quadratic growth has changed in recent times, but even nowadays, we are very much on a trajectory where the growth is very close to quadratic. So coming back to chirality here, chirality, and now coming back to, also to the possibility of life on Mars, chirality can in fact act as a very important biomarker because we should expect one of the two particular handednesses being present if life was responsible for the molecules or for the remains of that life. I remind you that the life that we have nowadays on Earth is in the form of carbonates, but the handedness has really been destroyed in that. That's simply because it's just pure graphite, and we can only see the biological functionality of former times in the isotope 13 being depleted. But if you want to look for life on Mars, we have to look at least for the possibility of life in the potential permafrost somewhere in the soil. So let me finally explain what kind of experiments have been done already back in the 70s. And this was a time way before anybody talked about astrobiology, the realization that life under extreme conditions can survive on Earth. And a very sophisticated experiment has landed on Mars successfully, and it was doing a number of different important experiments. And one of them is known as the label release experiment. It consists, it's very simple, and it can lead on experiments on Earth to the distinction or detection of life in desert formations where it would be with standard mechanisms rather difficult to find life. So it really uses soil, but also it lets it react with nutrients. Of course, this is here a picture of nutrients as we would like to have, but primitive life may actually survive on just very simple chemicals. And the question then is whether it produces any exhaust. Here, for example, in the form of CO2 or otherwise even methane in the presence of also heat and water. And indeed, the way that this experiment worked was by having the carbon from the nutrients being labeled. You do this by carbon 14 labeling of the nutrients. And those were nutrients that were supplied in the Viking experiments that brought these nutrients from Earth. But however, these nutrients were all, unfortunately, one must say, achiral. And so there was no detection possibility that one of the two chiralities could have been detected. The thing was, this experiment was in fact successful in the sense that it did produce CO2 because of the possibility. That's one possibility. If indeed the soil contained life forms that would react with the soil to produce CO2 in gaseous form. On the other hand, a control experiment was done by the same soil that was then heated to something like 50 degrees, originally more. And in that case, no CO2 was detected through measuring the radioactivity. The carbon-14 is radioactive. So these were important realizations, and here again, such curves from the actual lander of Mars, where you see that after something like 70 hours, you see a significant increase of the CO2 production, whereas in the control experiment, after you have sterilized the soil with just 50 degrees, no such reaction was possible. And this is not an easy thing to explain. With any chemical processes, even these chlorates that have been detected most recent or in the last 12 years, even chlorates that could in principle lead to oxidation and therefore CO2 production, why would they make a difference at 50 degrees? And this is still very unclear. And therefore, the this very interesting experiment was not explained at the time. And even nowadays, there's no good explanation for this experiment. And that's why one might think that one should actually repeat this important experiment in Mars. One idea was to do this with chiral organics that are being supplied as nutrients. And this was, such an experiment was done by a collaboration around Chris McKay, the PI on the Spirit and Opportunity missions back in 2004. He was still very interested in such an experiment. And what they discovered was that indeed, if you have glucose being used as a nutrient, you can supply both glucose of the L form, which is the non-natural, non-biological, biogenic form of sugar, and the D form of glucose. And here, in this case, we see that the consumption of the nutrients declines very rapidly if you take D-glucose, which is the glucose used in our world, and let it combust through eukaryotes. Whereas if you have glucose of the L form, those would not be used at all. Similar experiments have also been done with archaea and prokaryotes. And you see also a difference. The difference is not always equally big, but there is a difference. And it would be a very, very simple experiment to be done on Mars, even nowadays. And at least one should explain why the experiment back then was actually, in a sense, successful. The reason why the general community thinks the experiments were not actually successful is because no organics were found at the time. And the fact that no organics were found is very simply explained by the fact that, that we have these perchlorates, which leads to the destruction of any organics on Mars very, very easily. So you have to go a little bit below the surface and you have to be more careful, and then you actually do find organics. And that has only been possible in recent years. So with this, let me conclude by reiterating that the Viking experiments have in fact not been explained very much yet. That we do live with the possibility of the genesis of life being a ubiquitous mechanism that has to be proven. And so we must continue looking for the possibility, and even for the possibility of life on Mars. The handedness or chirality is in fact one important property of life. And as I've shown here, through the polymerization or the combined action of autocatalysis and enantiomeric cross inhibition, it could actually emerge. The handedness could emerge as a consequence of life. So the very first life was consisting just of some metabolism, and only one of the two has eventually started to lead to domination through various combinations of spatial possibilities and stuff like that. So we have to go back to Mars. We have to look also at high latitudes where we have the possibility of digging into the permafrost, where we may find perhaps also resemblance of extinct life, if the idea of extant life is not necessarily successful in the end. So with this, let me thank you for your attention, and let me open the floor for questions. So thank you.
All right. Thanks a lot, Axel. It was a great, very interesting presentation. A lot of things to cover. Now the floor is open for questions.
Axel, at some point in your talk, you introduced the notion of molecules with information.
Yes.
I wonder what that means.
What it means. So it allows for the possibility. I mean, once you have information, then you have then you can have the possibility of what is known as Darwinian evolution. So it would then know that one particular molecule could amplify more profitably in a given environment. It would lead to it would lead to evolutionary pressure. It's another form of mutual antagonism. It would lead to evolutionary pressure, and so only certain molecules that are fitter than others would lead to amplification. And this could lead to, for example, a change of from a early peptide world into, for example, an RNA world. And then finally, the next step would be, of course, that very primitive RNA molecules, which are not life as such, but they could perform some essential functions of life, including, for example, autocatalysis also is a possibility, or at least catalytic reactions. This was an important Nobel Prize discovery of Czech and Altman. So those molecules, they would evolve into other molecules that could undergo complicated chemical reactions, network reactions. And some of these reaction networks could be more profitable in producing and making one particular chemical apparatus or chemical automata, as they are called, be more profitable and more survivable under certain evolutionary pressure. And so in order to do that, you do need to have the information in the fact simply that molecules are different. And so it would lead to the memory of these molecules that can then lead to their preferential amplification. I hope that answers your question to some extent.
To some extent, perhaps I could pursue it with you offline.
Yes.
It's not clear to me what form this information takes, but I think you sort of explained it. Yeah.
Yes. So when it is simply the concept of Darwinian evolution that one has to have. So evolution that really reacts based on what this particular molecule or this particular information network actually is able to accomplish. So it's not just a single molecule that contributes to life, it's a combination of different molecules, and those have to be assembled. And in order to assemble this, nowadays life uses the information in catalysts through peptides or long peptide chains, of course, that contain the information, and they can lead to the particular formation of new RNA molecules through the proteins in which already and also contain the information. So the particular assembly of the mechanical assembly of the proteins, it contains information that in turn has its source in contemporary life from the underlying RNA, which led to its production. And so then these proteins again lead to the catalysis of particular new molecules and not any other ones.
Yeah, thank you.
Mm-hmm.
Pancha, you had a question. Go ahead.
Yes, this is Pancha Nawaz from the Faculty of Chemistry, and thank you, Axel, for the amazing lecture. And thank you for the disclaimer about chemists, of the relation of the light plane and chirality. So the way we chemists would like to think about the origin of life is that basically there are two aspects of it. One is the identity of all the chirality of all the amino acids, and the second is why it is the left one and not the right one. I was just wondering if you could comment on the relative contribution of these two factors in the current theories of origin of life. And the second question would be, if you have a life which is not based on carbon of other planets, then of course this postulates about the chirality being important would not necessarily apply. And so if you can tell us more about your thinking about these two topics.
Yeah, let me first answer the second question. Life not based on carbon has been discussed to various extent. One of the possibilities would be silicon, for example. Silicon is also has a valency of 4 and has some similarities, but it has by far not the possibility of developing complex enough chemistry. Another important argument is that in the case of carbon, we have the possibility of forming CO2, oxidated carbon. And that is a gas, and it can go into the atmosphere and propagate and connect other places. Whereas in the case of silicates, silicon oxide is basically soil, and that is not very mobile. So those are all various disadvantages to the possibility of life, which is not based on carbon. But those ideas, of course, have been discussed, and many people, astrobiologists, think that that carbon-based life is by far the more profitable one. But I have to ask you again about what you meant in the. So you are a chemist yourself, I understand, and so one of the things I wanted to say as a disclaimer back then in my talk was that the D and L forms do not one-to-one for all biogenic molecules correspond to dextrorotatory and levorotatory. That was what I wanted to emphasize for the chemists. But what exactly was your question? Can you repeat it again?
So I was wondering about the relative importance of all the amino acids being from the same chirality versus all of them being left. So this is just a very general.
In fact, the entire life could have been all the other way around, if that's what he meant. Is that what he meant? Yes.
Yes.
So indeed, that is an important realization. So all the ideas that are based on amplification mechanisms, which are the ones that I've been mostly working on myself, they would not make any difference between left-handed or right-handed life forms. Both would be equally probable, and it would be more than exciting to find a life form on Mars that had the opposite handedness. This would be extremely exciting. It would also prove of the idea that this idea of a model of autocatalysis and enantiomer cross inhibition may be at the source of this mechanism. The only reason why perhaps the left-handed life form, the left-hand amino acids could constitute life also elsewhere is perhaps because to some extent the weak force had an effect. And I did already mention the measurement of, or the idea that the weak force can lead to a preferential destruction of sugars of the wrong-handedness. And there was this important recent paper by Globius and Roger Blandford, and Naomi Globius is from NYU. Doing these experiments, it could be interesting. And there's another important measurement, and that is, I didn't come to that in my talk, but this is on the measurement of amino acids in meteorites. So many meteorites have been found, and there are some meteorites that also have a small excess of one of the two handednesses. And they work uniformly always in the same sense, that the left-handed amino acids are slightly more, by 2%, between 1 and 2%, more abundant than the right-handed ones. So if that is really true, and if that's really not due to contamination, which is another possibility, then there must be an explanation for at least that discovery, that meteorites and other bodies in the solar system show amino acids of preferentially one-handedness. And then the possibility of polarized photons could indeed become relevant to explaining that. And this could then give a particular weight in the early formation of one, namely particularly the left-handed form of amino acids at the early moment when the decision over life, over the origin of life was taken. So this could indeed make a slight difference and can then lead to the amplification. The subsequent mechanism that I described here would then simply lead to its amplification. So the early mechanism could really lead to a tipping tipping the pen in one of the two directions, and it would systematically tip it in one direction only.
Right.
That's all. Other than that, life, if everything is of the opposite handedness, would just function just perfectly. Of course, if somebody is able to manufacture proteins or meat, say, forming and for his follow-up label release experiments, if they really work out, that they of course demonstrate that metabolism